Good day everyone! Welcome back to my channel. So for today's lesson, we will going to talk about microbial genetics. So what is genetics? Genetics is a science concerned with the cell characteristics and how they are passed from one generation to the next. Gene is the unit of heredity. It is a segment of DNA that carries in its nucleotide sequence information for specific biochemical or physiological properties. Phenotype is all the heritable physical characters of the organism, such as eye color in human and resistance to antibiotic in bacteria. Genotype is the information of the DNA that controls the phenotype.
Main molecules of genetics are called nucleic acids. All the genetics information are stored as a sequence of bases through nucleic acids, mainly in DNA and RNA. In some RNA viruses, DNA, or stands for deoxyribonucleic acid, serve as organism's genetic material and divided into functional units or genes. Most of DNA is double-stranded, where the two strands held together by hydrogen bonds between A and T or G and C. The basic structure of DNA molecules is the nucleotide, includes phosphate, pentose sugar, and nitrogenous base. Prokaryotic DNA molecules are double helix and circular. These circular double-stranded DNA molecules are twisted and compacted through a process called supercooling or superhelicity. This is done naturally by topoisomerase enzyme. RNA ribonucleic acid is structurally similar to DNA, except most of RNAs are single-stranded. Sugar is ribose instead of deoxyribose, uracil base instead of thymine base. Genome is the total genetic information in an organism. Prokaryotic genomes, such as bacteria, consist of a single copy haploid circular DNA molecule, range from 580 to 4,600 kbp. Many bacteria contain extrachromosomal DNA materials as a part of genome called plasmid and transposons. Eukaryotic genome, such as fungi, is carried into two or more linear chromosomes separated from cytoplasm by nuclear membrane. Diploid eukaryotic cell contain two homologous copies of each chromosomes. Eukaryotic cells contain mitochondria and chloroplast, and some eukaryotic, like yeast, contain plasmid. Eukaryotic genes, unlike prokaryotes, are interrupted by introns. Viral genome, unlike others, may be of DNA or RNA. They are capable of survival, but no growth in the absence of host cell. Replication of viral genomes depends on the metabolic energy and the synthetic machinery of the host cell. DNA replication is that one DNA molecule gives two DNA molecules. Each one consists of one strand from the original DNA, and the other strand is newly formed one. Replication starts at a fixed point called origin of replication or ORIC and terminated at fixed point called TER, point step of replication. There are 5 steps of replication. Step 1. The two old strands of DNA are separated by helicase enzyme to form the replication fork. Step 2. Replication of one strand template in 5' to 3' direction can proceed in a continuous fashion starting by addition of short segment of small RNA at the starting point called primer. On this primer, DNA polymerase enzyme start to synthesize the new strand in a continuous manner from 5' to 3'. This is called the leading strand. Step 3. Replication of the other template strand can only proceed continuously. This strand is called the lagging strand. Many primers or RNA primers are used to synthesize the lagging new strand as a fragment called Okazaki fragments. Step 4. Then all the RNA primers are removed by the DNA polymerase 1 and the gaps are sealed by DNA ligase to make continuous DNA strand. And step 5, after formation of two strands of DNA, the old one and the new one DNA carries enzyme or topoisomer to make twist and superhelicity of each DNA molecules. Gene expression or protein synthesis is the mechanism by which the sequence of nucleotides is gene determine the sequence of amino acids in proteins. Occurs in the following steps: transcription and translation. In transcription, the DNA sequence of gene is copied to make RNA molecules. This step is called transcription because it involves rewriting or transcribing the DNA sequence in a similar RNA alphabet. Transcription have three steps such as the initiation, elongation, and termination. In initiation, ribosome assemble at specific AUG of mRNA and binds to tRNA AA, two codons at a time matching complementary anticodons with mRNA codons. In elongation, ribosome catalyze peptide bond formation between amino acids attached to each tRNA and shift 3 nucleotides 1 codon on mRNA and repeat the process in termination. Stop codon causes translation to an end. Gene transfer refers to the movement of genetic material. There are two types of gene transfer: the vertical gene transfer and the horizontal gene transfer. In vertical gene transfer, the transfer of genetic material is from parent to offspring. It may be through swapping of genetic material between neighboring contemporary bacteria. Is another means by which resistance can be acquired. Many of the antibiotic resistant genes are carried on plasmids, transposons, or integrons that act as vectors that transfer another genus or species. Horizontal gene transfer may occur via three main mechanisms: transformation, transduction, or conjugation. Transformation involves uptake of short fragments of naked DNA by naturally transformable bacteria. Transformation involves uptake of short fragments of naked Transduction involves transfer of DNA from one bacterium into another via bacteriophages. Conjugation involves transfer of DNA via sexual pilus and requires cell-to-cell contact. DNA fragments that contain resistant genes from resistant donor can then make previously susceptible bacteria express resistance as coded by these newly acquired resistant genes.